Good morning students, welcome to LEELA'S tutorial. In today's class, let us discuss about modification and isolation of desired gene of interest. Right? So here in the livestock, we want to introduce a beneficial gene so that the animal will be performing better. Right? So how you are going to select that gene, how you are going to isolate that one, and how you are going to modify the gene, that we will be discussing here. So in the first, we will be seeing for the isolation, right? Before modification, let us go for the isolation of the genes. That is the desired gene. That means here the desired gene is not present in that particular animal. You have to take the gene from the another host animal, right? First you are going to check for the desired qualities which you need. Then that animal will be selected and its DNA will be collected. So what is the process? First it is isolation of the gene of interest. For that you are going to select the host animal and collect the cells. Then from the cells you are going to undergo the cell lysis process so that the genome, the nucleus will be out. Then from that nucleus again by various steps you are going to isolate the DNA. Right? So here you have only the DNA but not any cell organelles or the cell debris, anything. So now you have the purified DNA, and this purified DNA you require only certain region or certain fragment which consists of the desired gene of interest. Right? So you don't want the complete DNA or the complete genome of that host animal. So what you are going to do? Here you are going to select the specific probe for that desired DNA. So here you have the desired DNA region, so where the specific probes are added. So here that probe will be helping us to identify the particular location of that desired gene of interest. So now once you have done this, now you will be adding the restriction digestion enzymes, so that only the required region will be sliced, excised, and you will be getting the desired gene of interest. Right? So now this desired gene of interest can be introduced into the host animal. Okay? So here we have given, first you are going to collect the cells, then undergo cell lysis, then isolation of DNA, then selection of specific genes using DNA probes. Right? That is your isolation of the desired gene of interest. So now let us see how you are going to modify it. So it is modification of the genes, which is also referred as genetic modification or genetic engineering process. Right? So now the genetic modification is of two types, the mutagenesis or recombination process. So let us see the mutagenesis first. So in mutagenesis you have various types. First one is gene addition or insertion, gene deletion, gene substitution, gene translocation, inversion and all. Right? So in this case you can modify only that particular region where the gene has to be present or replaced, or it can be substituted. So let us see the gene addition here. Look at this picture. So here in the gene addition or gene insertion, in the chromosome you have A, B, C and D. Right? So here we want to change the base so that the gene will be modified. Right? You have A, B, C, D. Right? So here in this base addition, you can see we have added the Z. Right? So that means here we have inserted or added the desired gene of interest. Right? Then in second case, another mutagenesis, it is deletion. So here you can see one of the base has been deleted. Here you have A, B, C, D over the chromosomes, but here A has been deleted. A has been deleted in this step. So it is base deletion. Then in next one, it is substitution. So in case of substitution, again you can see A, B, C, D on this. How it is modified? It is instead of B region, right, the B instead of B region, you have replaced E. Right? So here this is base substitution. Then the next one is translocation. So in the translocation, you can see that A, B, C, D genes here. The A gene has jumped. Right? It has been translocated to another gene E, F, G, H. As a result, that gene is modified with A, E, F and G, H genes. Is that clear? So it is translocation, whereas here in these cases within the chromosome there is a modification within the chromosome, but here the gene has moved from one chromosome to the another, or one place to the another place or the loci, right? Then the last one, it may happen naturally also in mutagenesis. It is A, B, C, D. You can see there is overlapping of the chromosome, that is inversion. Right? Because of this inversion, instead of A, B, C, D, you can see A, B, D, C. Is that clear? So, when it is open, now it is somewhat coiled, inverted. So, when it is open, there will be exchange of the bases. Right? In such a way, you can modify the DNA or modify the gene of interest. Okay? Then coming to the recombination. So here the recombination itself implies that there is combination of two chromosomes. So here the first one, you can see the selection of the donor. First you have to select the donor animal from which you want to isolate the desired gene of interest. So second step is isolation of desired DNA. Then it is insertion of the desired DNA into the vector molecule. So here you are not introducing directly into the animal, but you are making use of an vehicle, that is the vector molecule or the carrying vector. So then this carrying vector will be indirectly introduced into the host animal. So here you can see in the diagram, once you are selecting the desired animal, the donor animal, its DNA, again the cell lysis, then DNA is isolated. After you isolate the DNA, this is the DNA. After you isolate the DNA, only this region is the desired gene of interest. So here by means of restriction enzymes, you are going to cut or restrict the DNA and isolate only the desired gene of interest. And this desired gene of interest will be inserted into an vector molecule. So this is your desired gene of interest which is introduced into the vector molecule. Then this vector molecule can be introduced into the host organism. Otherwise, directly the desired gene can be injected into the oocytes of the host organism by means of micro injection process. Right? Already we have discussed that technique. So where the directly the desired gene can be injected by means of micro injection or micro manipulation technique into the host oocytes. Right? So in such a way you can modify, you can select the desired gene of interest and also modify the genes and then introduce that into the host animal. Is that clear? Okay. For further videos, subscribe.